Okay, so this is a chemistry video for biology. So biology is the study of life, but biology is really just applied chemistry, because all of biology can be explained using the chemical functions that are going on in life. So knowing that biology is just applied chemistry, there are a few basic concepts. One of them is carbon, one of them is water, H2O, and then the other one is DNA, which stands for deoxyribonucleic acid. It's a really long word. It's not all that important to know, but the key is that DNA is genetic information for all life. Genetic information is the heritable or the passed downable genes that end up determining what something looks like or what something does. Also, DNA could be, there's RNA, which sort of complements DNA, which is ribonucleic acid, so there's one fewer oxy, so it's, rather than dioxy, it's one oxygen group. And RNA is the main heritable material in some prokaryotes and in some life forms with less going on in them. So now, in the chemistry of life, the important thing to know about chemistry is everything's made up of matter. Matter is just, if something exists, there is matter in it. Now, all matter is made up of elements, and the elements can be found on the periodic table. The periodic table looks sort of like this. There's like a weird thingamajigger. You've probably seen it in your chemistry classroom. You got your non-metals, you got your metals. Most of these are gases up in there. But the really key thing is that all elements are made up of protons, neutrons, and electrons. Protons have a positive charge. You can remember because positive and pro. Neutrons are a neutral charge. You can remember because neutron sounds like neutral, so 0. And electrons have a negative charge. And you just have to remember that because it's the only one left. These three building blocks have to do pretty much create every single thing inside the cells. Protons determine what element it is, electrons determine how charged that element is, and neutrons determine how heavy that element is with the protons. Protons have an atomic mass unit, AMU, of 1. Neutrons have an AMU of 1, and electrons have an AMU of close to 0. I think it's 0.8 or something. It's essentially 0. So we don't count electrons when we're coming up with the molar mass of a compound. So now, beyond that, there are three. The important thing to know is that elements will then bond. Elements bond into compounds and molecules. And there are three big important types of bonding to know for biology. One of them is ionic. One of them is covalent. And one of them is hydrogen. Hydrogen is abbreviated by the letter H. It's a good element to know. Carbon, abbreviated by the letter C. Oxygen is O. Those are the big three you must know. Ionic bonding happens between a nonmetal and a metal. It happens with an element on one side of the periodic table and the other side of the periodic table. And it happens when an element actually donates an electron to another element. And then, because this element has donated an electron, it lost a negative charge, so it became more positive. Because this element gained a negative charge, it became more negative. And so those two are attracted just like a magnet, and those two are bonded. Now, covalent is when two nonmetals bond, and they actually just share electrons. So there are no dipoles. And so a dipole is this positive or this negative. And so covalent bonds are usually stronger than ionic just because they're sharing electrons rather than being attracted like that. Hydrogen bonding has 100% to do with dipoles. Hydrogen bonding is very similar to ionic bonding, except it only is between two molecules and only between the hydrogen and another element in those molecules. A good example of this is water. Here's my O, here are my H's, and then here's another water molecule. Well, we know that water exhibits hydrogen bonding within itself. And I'll explain that a little bit later when I get to hydrogen. But the reason why is because this O has a bunch of electrons around it, and this H does not. If you look, the number of valence electrons for the oxygen is 6. The number of valence electrons for hydrogen is only 1. So the O is a lot more negative, so we mark it as a negative dipole. This is the symbol for dipole. The H's are a lot more positive, positive dipole. Well, now you can see this positive dipole right here will be attracted to the negative dipole right there. And that's what causes hydrogen bonding. Hydrogen bonding leads to some pretty amazing properties of water. Now, water is a polar molecule, which is how hydrogen bonds. Here's a great picture of some water. It's just H2O. And so when water is bonding to itself, like this, using hydrogen bonding, we call that cohesion. And co is the prefix for with or together, so it is bonding together with itself. Now, adhesion is when water bonds to other things. When those two combine, they create capillary action. Now, capillary action, let me just erase this for a second. Capillary action is what makes water travel up tubes. Well, so the water molecules at the bottom, here's my water molecule, is attracted to another water molecule, but it's also attracted to this little particle right here that's part of the tube. So it gets pulled up, and this pulls all the other water molecules up, and they travel up the tube. And this is how trees, excuse my shoddy picture of a tree, there's a little swing on the tree. That's how trees get water to the highest leaves. Now, besides capillary action, water is really important because it has a high specific heat. It's not really important to know 100% why water has a high specific heat for biology, but it's important to know that the high specific heat means it's really hard to change the temperature of water. The specific heat is the way we measure the hardness, or how much energy must go into changing the temperature by 1 degree. And water has a really high specific heat, so it's really hard to change the temperature of water, which means it's hard to freeze water, it's hard to boil water. This is what keeps our bodies working using homeostasis. We stay at the same temperature because the water will heat, but it won't boil, and it will cool, but it won't freeze. Heat of vaporization and heat of formation are considered the heat. Heat of vaporization is the boiling temperature, and the heat of formation is the freezing temperature. But for water, those are very far apart. Water, let's say water's naturally right here at about room temperature. It's really hard to heat it, so it's boiling, and it's really hard to cool it, so it's freezing. A lot of other elements look more like this, where it's not that hard to freeze it, and it's not that hard to heat it. So this big distance in between the boiling point and the freezing point of water make it a really important part of life. Also, water is the universal solvent. So if I create a solution, which is just when I mix water, or when I mix two chemicals, they form a solution. The liquid in my solution, the thing that I'm mixing the solid into, is the solvent, and the solid that I'm putting into the solvent is the solute, and those form the solution. So water is considered the universal solvent because almost everything will break apart in water. If you've ever tried to put if you've ever tried to put salt in water, the salt actually seems to melt into the water, and that's the water is breaking down the crystalline structure of the salt, and it's acting as that solvent, and it forms salt water. A key thing about water is it also acts as a very weak buffer. And a buffer it keeps the pH very stable. So in a solution, if you insert an acid or a base, the pH will change a lot. But if you insert a buffer, that will sort of keep the pH from changing by adding ions that otherwise would have been taken out so that they don't change. It's not all that key for biology. Just know that you've heard the word buffer and sort of understand. Just think buffer keeps pH the same. You don't need to know that much more. Okay, last big topic is carbon. carbon is really key because carbon is the foundation of life. Biochemistry is just the study of how we use carbon in our bodies and how carbon travels through the world. Important things, organic chemistry is all about studying hydrocarbons. Hydrocarbons are hexagons, usually, with carbons at all corners, and then various other bonds outside. These are also called benzene rings. Sometimes they have double bonds. But it's just important to know that if you see this, you know you're dealing with an organic molecule, and you know you're dealing with biology rather than chemistry. Now, so carbon forms a few, so carbon is the basis of organic chemistry, and in organic chemistry there are really two main important things to know for biology. One is isomers, I'll spell that out, isomers, and then the other is functional groups. And I'm going to go over the isomers and functional groups, and then you know all the chemistry that you need for biology. Isomers, there are three types. There's a structural isomer, and a structural isomer is when something has the same formula but a different shape. When I think of structural isomer, I like to think of two different types of pickup trucks. I like to think of a Ford and a Chevy. They've got the same formula, they've got the same parts in each, but they don't look the same because they've been arranged in a different way. So there you go, that's a structural isomer. Then you've got a geometric isomer. And geometric isomers have the same formula, but a slightly different orientation. So really, the most common thing you'll see for a geometric isomer is you'll see a C bonded with a C, you've got element X over here, and you've got element P over here, let's say. Well, this we consider the trans isomer, because these are across from each other. And this, when they're next to each other, C, C, P, P, we call this the cis isomer. And that's just something that you won't see a lot of isomers on the AP Biology exam, you won't see a lot of isomers in biology, but it's good to know the difference between a cis and trans isomer and that there are geometric isomers. The last type of isomer is a enantiomer, which is a long word. This one is the least important of the three, and the easiest way to remember this one is your left hand and your right hand. They've got the same formula, they've got the same structure, but they look slightly different just based on how they're twisted. So a left-handed isomer, if I show you the palm of my right hand, it looks identical in terms of fingers to the back of my left hand. And so you can see that they seem identical, but if I show you both palms, they're clearly different. And so that's what we would consider an enantiomer, And a good example of that is L-DOPA. There's L-DOPA and R-DOPA. L-DOPA is an effective drug. R-DOPA is actually poisonous. So you need to make sure you get the left-handed version of DOPA if you ever need it. And so those are isomers. Isomer means it has the, wrap-up isomer means it has the same chemical formula but a different shape. Okay, final thing, functional groups. I'm going to do these in order of importance. The first most important functional group is hydroxyl, OH. Now the hydroxyl makes alcohols. Hydroxyl is also what we consider a base. So if you see KOH, that's a base. And so hydroxyls are really important. Then we've got carbonyls. Carbonyls are just a C and an O, and those split into ketones and aldehydes. You really don't need to know much of the difference between ketones and aldehydes, but it doesn't hurt. You can look at that yourself if you're interested. Then there's carboxyl. So this is carbonyl, and this is carboxyl. Those are key to keep separate. And the carboxyl is just a combination of the hydroxyl and the carbonyl. Next, we've got the amino group, or the amine group, which is an NH2 group. And this makes amines. These are really common in life because there's a lot of nitrogen in our bodies. And so you'll often see the NCC. And if you see that N, it's usually got two H's off of that. And those two H's are that amine group. Second to last, you've got sulfhydryl, which is SH, which is sulfur and hydrogen. Not that important. Honestly, I never saw it in AP Bio. Don't need to worry about it. And the very last one is phosphate, PO4. And phosphate is important because it makes organic phosphates. And the organic phosphates are what we use as energy. And so, if you've ever heard of ATP, that's a terrible A, or ADP, that stands for adenosine triphosphate. So we've got an adenosine with 3 triphosphate groups on it, whereas this one is an adenosine diphosphate. We've got an adenosine with 2 phosphate groups on it. And this one provides us energy, and then becomes that one. And this one can become re-energized and become ATP. So the phosphate groups are what provide energy in the form of the electrons and the charge stored in them. Beyond that, it's just really important to understand that of all the things that I mentioned, really the key ones are ATP, ADP, DNA, RNA, and hydrogen bonding. Hydrogen bonding comes up so often that if you're not sure about the answer and it seems like it's a chemistry question, hydrogen bonding is usually your answer. Thanks.